Hello class, how are you doing? Today I'm going to go over chapter 10, and chapter 10 is about this leaf photosynthesis. Okay, we'll talk about photosynthesis for this chapter, and then that would be the end of the material for your next exam, which is exam number 2, exam number 3 anyhow. Whatever exam it is, I'm losing track of things, but anyhow, that's okay. Next exam, that's the end of it. So the process that feeds the biosphere, yes, we are depending on photosynthesis heavily, is a process that converts solar energy into chemical energy, and directly or indirectly photosynthesis nourishes almost entire living organisms. Autotrophs are organisms that make their own food. Right here, they make their own food. Heterotrophs are organisms like us. We depend on autotrophs or other heterotrophs. We do eat animals. We are omnivores. And then, so that's what heterotrophs are. It means hetero, it means different. And auto, it means self. Those of you have it on your prefix suffixes, self, and hetero, it means different. And troph, it means nourish, right? You know that trough, the word trough, you're familiar with that, means nourish. So when we are using other organisms, that's then we are heterotrophs. Okay, so having said that, let's move on. What is the Earth's supply of fossil fuels was formed from the remains of the organisms of Tenia. Intensity of fossil fuels represents the solar energy. So you can read all of that and enjoy it. Photosynthesis converts a light energy to chemical energy of food. And what is anatomy of chloroplast? That's what I'm going to talk about next. This leaf, I keep losing it, is made up of, why you seeing it green? That's another thing we will talk about. So they are mesophyll, which is the middle layer of leaf, and each mesophyll cells contain 30 to 40, about 30 to 40 chloroplasts. Carbon dioxide enters and oxygen exits from leaf, and microscopic pores is called stomata. So this leaf, there are little pores in here, if you would. These little pores are referred to as stomata. Okay, you cannot see it. When we cut it and put it on the microscope, then we can see it. They are microscopic. You do not need electron microscope to see it. Of course, you can see it with electron microscope too, but they are microscopic. Here they are. So here is a leaf. The middle layer, right here, from epidermis to epidermis, that middle layer is called mesophyll. Okay. And then these openings are, the veins are made up of xylem and phloem. I'm not going to talk about it. This is not a botany course, but you should know xylem and phloem. Xylem and phloem. Okay. Phloem moves food, and xylem moves water. That's called vascular bundle. They call it vein. But anyhow, and then of course you can see the stomata right here, that the carbon dioxide needs to go in to make glucose molecules, and end product oxygen comes out. We will talk about that as well. So I did take a picture of the chloroplast for you in the lab. This is a chloroplast, and these are the anatomy of it. The parts of the chloroplast you should know. Each one of these stacks, you cannot see it on the microscope anymore. You can see the chloroplast on the microscope, but each one of these stacks is called thylakoid. Okay, these are all thylakoids. So when stack of thylakoids is on top of each other, it's called granum. Okay, and then when you have a few granums are attached to each other, it's called grana. Okay, and the medium. You remember in mitochondria, the medium of mitochondria was called matrix. The medium of the chloroplast is called stroma, right here, okay? Do not confuse that term, stroma. There's an R in here. you guys look, there is an R in here. Do not confuse it with the term stomata. Stomata are the pores of the leaf, but stroma is the fluid, the medium inside of the chloroplast, equivalent to matrix, equivalent to cytoplasm in a cell. So the medium, remember, this is membrane bounded. There are 2 layers of membrane that bounds chloroplast. And then, of course, you have the inner membrane and then outer membrane. We talked about this. And then with the mitochondria, but here they have inner membrane, outer membrane. And chloroplast is an envelope of stroma. Okay, we talked about it. Dense fluid surrounded, it's called stroma. Thylakoid are each stack and connected stacks, the chloroplast, which compose the 3rd membrane system. And the chloroplast is, it has itself a membrane, and then I grind them for you in the lab. The thylakoids may be stacked in a column. It's called granum, singular grana. They're connected together. And chlorophylls, they are pigments which are in the chloroplast. And in the lab, I did separate them for you. I grind the leaf, the spinach leaf, and then you saw it. Okay, how to track what is that? I misspelled like it. Track atoms through photosynthesis, scientific inquiry. Okay. Photosynthesis, as you know, this is the formula for photosynthesis, that it has been balanced: carbon dioxide plus water. You already had chemistry, so this has been balanced for you guys. So, and it gives you light energy plus glucose molecule, oxygen, and of course it gives you water. This is an endergonic reaction. It's storing the energy. It's opposite of entropy. Okay, I do not have a term for it, but anyhow, the overall chemical reaction during the photosynthesis, reverse of that, the one occurred in the cellular respiration. You remember during cellular respiration, in the last chapter, we talked about this glucose molecule with help of oxygen in the mitochondria. It breaks down eventually all of those carbon dioxide that comes off of the Krebs cycle, and also comes off from acetyl-CoA is converted to pyruvate is converted to acetyl-CoA. Okay, all of that. And then, of course, I never talked about it, that formation of the water, and then it gives you ATP at the end. ATP and NADPH. NADP, not NADPH. You will see NADPH here in photosynthesis. Here it is. They've done an experiment that they wanted to see, if I ever ask you, you should know this, that where does the oxygen of photosynthesis comes from? This oxygen. That's the question. Okay, what happened? Scientists, they made the carbon dioxide radioactive, and when they made carbon dioxide radioactive, the carbon, of course, it appeared in glucose molecule, and the oxygen of the carbon dioxide was incorporated in a glucose molecule. And the water is also another end product of photosynthesis. The water molecule was also, the oxygen in the water molecule came from the oxygen of carbon dioxide. However, for photosynthesis, you do need water. Right here, the water of the oxygen of the water, showed up as the oxygen that is emitted into the atmosphere by plants. Okay, so that is an experiment they've done. They call it tracking, and then pasting, tracking. What do they call it? I'm forgetting nowadays. I should have wrote it down. It's called tracing and tracking. So let's go back. What they've done, they just like birds, you know, they put a little tag on birds. It transmits waves, and then you can follow where birds are migrating, where they are going. Same thing they do it in biochemistry. They tag Okay, they call it labeling. Label. They label carbon, oxygen, whatever, with radioactive material so it can glow, it can show. And then after they label it, they trace it where it ends. Okay, so when they trace it, that carbon, of course, ends up in glucose molecule. That oxygen, after they label it in glucose molecule, and that oxygen also appears in water molecule as well. Okay, photosynthesis reverses the direction of electron flow compared to respiration. Photosynthesis is the redox process in which water is oxidized and carbon dioxide is reduced. Okay, so these are the things I would like you to know. Photosynthesis is an endogenic reaction process, which I talked about that. It was provided by energy of light. Here it is, what is being become reduced, what carbon dioxide is being reduced, and then it becomes oxidized, water, the one that is becoming oxidized. Okay, I hope that is something you should know. The two stages of photosynthesis, important, very important, the light reaction and Calvin cycle. When I was a student, they used the term light independent or dark reaction, and that was very confusing for me. What I was confused with, that dark reaction happens only at night during dark time. That is not true. That's why they changed it to Calvin cycle. Okay, so the students like me who are confused will be straight. So Calvin was the name of the scientist first discovered the Calvin cycle. So it's not being called dark reaction anymore. So light independent reaction. That's independent reaction. So during light reaction, you should know that water molecule is being split. And then it releases oxygen, and then reduce the electrons accepted to NADP becomes NADPH. We did not have, in cellular respiration, we did not have NADP. We had NAD and NADH, okay? The only difference here is a phosphate. They have a phosphate attached to it. And also generate ATP from ADP, which you'll see it here in a minute in the photosynthesis. The Calvin cycle, the light independent, the cycle that you will see in the diagram, that it does not need energy, light energy. Here in this, in the light reaction, you do need, and it happens in thylakoid. Of course, you should know where, just like cellular respiration, you know, glycolysis occurs in cytoplasm, and then the electron transport chain and the Krebs cycle occur in mitochondria. You know those. Okay, same thing here. You should know that the light reaction occurs in thylakoid, and then the Calvin cycle happens in the stoma, which I will go over it here in a minute. Hang on. So the Calvin cycle forms sugar by using the carbon dioxide, of course, from the air, and using ATP and NADPH. They are high energy molecules. These two are high energy molecules. The Calvin cycle becomes the carbon fixation. Okay, make sure you know that term, incorporating carbon dioxide into organic molecule right here. So this is the light reaction. What is happening? Water is coming down into thylakoids, stack of thylakoids, granum, singular. And then, of course, you know, Chloroplast has 2 layers of membrane right here. You do know that. And then what happens, the NAD becomes NADH. NADH2, okay? And then ATP becomes ATP. And then these two are being used here. I think it comes up next. And then, of course, when water molecule is broken down, some some of that oxygen goes into the atmosphere, get released by the plants. Okay. As I said, ATP is made, NADPH is made also. I'm sorry, not NADPH2, sorry about that. NADPH is made too. So NADPH and ATP will go through the Calvin cycle, okay? And in the Calvin cycle, what happens, of course, CO2 comes in, and then from Calvin cycle, you get these two guys, and then what gets out of Calvin cycle is glucose molecule. Okay, so right here is the Calvin cycle, carbon dioxide comes in, as I said, NADP, NADPH becomes NADP, ATP becomes ADP, and of course they draw you the glucose molecule. This is a complete version. This is what you should know. Oh, of course, you should know that the Calvin cycle, the light independent cycle, occurs in the stroma, in the fluid portion of the chloroplast. And then, of course, the light reaction occurs in the thylakoids. All of this happens in the chloroplast. Everything is happening in the chloroplast. But two separate cycles, light and dark. Okay, I'm sorry, Calvin cycle. So the light reaction occurs in thylakoid, and the dark Calvin is a habit, I guess, from my freshman, sophomore year in college. But the Calvin cycle, it happens, the light independent cycle, Calvin cycle happens in the stroma. This is not significant, guys. I saw this. This is not that big of a deal. It's telling you that how animals and plants are depending on each other. These animals quickly are eating plants, vegetation, and then other animals like carnivores, they eat these animals. So these animals, the carnivores, are indirectly depending on photosynthesis. If photosynthesis was not occurring, these animals would die, and eventually when these animals will die, the herbivores die, the carnivores would die too. That's all the carbon cycle is talking about, so carbon cycle. And of course, when these animals die, they are on top of food chains. They release carbon into the atmosphere. They release food and nutrient here. And these guys, the plants, will take them as a nutrient, as a source of food, and the cycle goes on and on and on and on. So I'm not going to show it to you guys. Now this one is important. Let's watch this one.
Plants provide us with food to eat and oxygen to breathe. They perform this amazing feat by the process of photosynthesis. Let's take a closer look. Photosynthesis requires carbon dioxide, which diffuses into the leaf through small pores and then enters the cells. Inside the cell, carbon dioxide diffuses into the chloroplasts, where photosynthesis takes place. Chloroplasts use energy from light to transform carbon dioxide and water into sugar and oxygen. Zooming into a chloroplast, we see these flattened membranous sacs called thylakoids. Here, light energy is converted to chemical energy in the first phase of photosynthesis, the light reactions. The two green structures you see here are photosystems, large complexes of proteins and chlorophyll that capture light energy. An electron transport chain connects the two photosystems. Okay, notice the small mobile electron carriers that shuttle electrons from one large complex to another. Now let's take a closer look at the steps of the light reactions. The photosystem on the left absorbs light energy, exciting electrons that enter the electron transport chain. Electrons are replaced with electrons stripped from water, creating oxygen as a byproduct. The energized electrons flow down the electron transport chain, releasing energy that is used to pump hydrogen ions, the blue balls, into the thylakoid. In the photosystem on the right, light energy excites electrons, and this time the electrons are captured by an electron carrier molecule, NADPH. The high concentration of hydrogen ions inside the thylakoid powers ATP synthase, producing ATPs. The light reactions in the thylakoid have produced two energy products, ATP and NADPH, that will now power the production of sugar in the Calvin cycle. The Calvin cycle takes place outside the thylakoids in the stroma, the thick fluid of the chloroplast. At the beginning of the cycle, carbon dioxide molecules combine with molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light reactions. Sugar molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin cycle again. But the important product of photosynthesis is the remaining G3P sugar. Some G3Ps are used to build glucose, which can combine into starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration using oxygen in the plant's own mitochondria, generating ATPs that can power other work of the plant. Excess oxygen diffuses out of the leaf through the pores, while more carbon dioxide enters. With three simple ingredients, carbon dioxide, water, and light, plants produce sugar and oxygen by photosynthesis, powering plant metabolism and ultimately providing your fuel as well.
Right. He pretty much said, she talked about the photosystem 1, photosystem 2, which I will get into it in a minute. Here's a light spectrum. If you pass the light, I need to talk about that too. If you pass the light through a prism, I don't know, a pyramid-shaped prism you had it when you were a child, this is what it flashes up, okay? That's what flashes up. And then what happens, the plants that are green, okay, they are reflecting this end of the spectrum and they are absorbing everything else, okay? So that's why you see plants green. Most plants are reflecting green because of that. And then other plants that you see red or purple or yellow, they are reflecting those. Okay. Here is the, we talked about this, the light, it comes into photosynthesis, it comes into chloroplast, the thylakoid, and what happens, release light, and so on and so forth. There's another animation. Let's watch this one.
The thylakoid membrane in chloroplasts contains light-absorbing molecules called pigments. Different pigments absorb light at different wavelengths.
Right. I haven't talked about
One
this.
pigment, chlorophyll A, absorbs mainly blue-violet and red light. This molecule participates directly in the light reactions. Another molecule, chlorophyll B, absorbs mainly blue and orange light.
Right.
We do not see these absorbed colors when we look at a leaf. Instead, we see the green wavelengths that are reflected back from the leaf or transmitted through it.
Okay, so let's talk about different type of chlorophylls. I think that's next. A spectrophotometer measures the pigment's ability to absorb various wavelengths of the light, and that's what I did in the lab. Used to, we cut off those different type of chlorophylls and pass them through a solution and put the tube in a spectrophotometer machine and see how much light is being absorbed. Okay, the machine sends a light through. Here is, do not worry about that slide. I'm not gonna ask you anything about the spectrophotometer machine, but that's how it works. Okay, this is important. I would like you to know. What are different type of chlorophylls and their functions? So if I have two containers, a pie flask as such, okay, I want you to answer this question. And then one of them, I have a tube in there, and the other one, I have a funnel like this, and I have a beaker full of water. which one of these two is more efficient way, A or B? There's no brainer, but I always ask that in the class. So is more efficient way to put the water in container A from this beaker or container B? Which one you think is more efficient way? This has a funnel, okay? And this has just a tube, straw. Of course, B. B is more efficient, faster. It takes all of that water and dump it down here. But here, it's very slow. You might spill some water here and here, but B, all of that water goes inside of the Erlenmeyer flask. Same thing happens in photosynthesis. Okay, photosynthesis, chlorophyll A, right here, as I said, participate in photosynthesis directly. So this right here, that would be chlorophyll A, okay? Then, of course, they write them down small a, small b. Chlorophyll B and carotenoid and xanthophyll, they are three major type of chlorophyll. They absorb light right here. They absorb light right here, and they give it to chlorophyll A. I hope I'm making some sense. And that's why the system is more efficient to absorb light. These guys, the other wavelengths, which they do absorb other wavelengths of light. Where are you? Where are you? Right here. Other wavelengths of light. Okay, they absorb these and they give it to chlorophyll A. Right here. Okay. I hope I made some sense. So here they are, the light absorbance of chlorophyll A, chlorophyll B. I don't expect you guys to memorize those numbers. You're not taking botany. I'm not sure even in botany they ask you to memorize those numbers. I'm not too crazy, but you should know chlorophyll B, carotenoid, and xanthophyll. Right here, I list them for you all. Carotenoid is like orange color, and then xanthophyll is yellow, and chlorophyll B is a kind of like a blue-green. Here is the structure of chlorophyll A and chlorophyll B. So again, the main thing is that methylene right here, it's they call it porphyrin. Is that right? Porphyrin ring. That's called this ring is called porphyrin ring, which is magnesium is inside. Light absorbs head molecules, note magnesium atom in the center, and then that is a hydrocarbon tail. And chlorophyll A has methane, and chlorophyll B has a CHO structure right where it's supposed to be. Okay, now we are talking about photosystem 1, photosystem 2. That's what the last, they talked about it. Again, the great details of this, I don't expect you to know. I don't want you to spend too much time as long as you have general ideas of photosynthesis, then you should be okay. What happens with the light, this is a photosystem 2. First, most textbooks explain photosystem 2, and then they explain photosystem 1. What happens during the photon from the light comes in, and then what happens, it activates some electrons, and these the photosystem harvests light. The photosystem 2, as I said, again, you should know photosystem 2 right here harvest light, and you should know that. And of course, it is in the thylakoid. There are two types of photosystem in the thylakoid. Photosystem 2, PS2, functions first. Numbers of reflected orders are discovered that best absorbed by wavelengths of 680. Again, I'm not too crazy about that number, but you never know. The reaction center of chlorophyll A of the photosystem 2 is called P680. Okay, so that is, so you remember chlorophyll A participate in the photosynthesis directly. My funnel Let me go back. My funnel right here. Okay. So what he's saying that photosystem II contribute. What is photosystem 1? Is the best absorbing wavelength of 700 nanometer. Okay. So the other one, photosystem 2, is 680. This one is 700. The reaction center of chlorophyll 2 of PS1 is called as P700. So this is, we already saw that. I'm not gonna say anything about it. Let's move on. The photosystem 2, as I said, is P680. Okay, well, he's referring to it a lot. So maybe might as well, let's say, you should know it. So the light activates these pigment molecules, right? And then what happens next, of course, the water molecule breaks down during this system. You should know that the water molecules break down in the photosystem 1, and then oxygen is released, and the hydrogen ions move to the next step, which puts up the cytochrome complex, which release ATP, electron transport chain. The electrons are being transported to cytochrome complex, and cytochrome complex makes ATP. And one of the cytochrome complex is ATPase. Okay, so ATPase is involved here in order to make ATP. Okay, then what happens again? Photosystem 1, you have this is all light. These are all light dependent structures, you guys. Photosystem 1, what happens? The light comes in, and then of course you have the P700 involved, and primary acceptors of electrons and so on and so forth occurs in P1. And then during that stage, NADPH is made. Okay, NADP becomes NADPH. So again, you should know this. During photosystem 1, NADPH is made. During photosystem 2, ATP is made. Okay, important. You should know those. I'm not too crazy about the electrons here and what happens, details, but the gist of it, NADP reductase is the name of an enzyme, converts NADP to NADPH. And that enzyme complex is a complex of enzymes. It gets its electron, it gets its energy from, of course, the light energy, and then the other. Okay, so this is the diagram showing you how the electrons, one by one, they can help and build ATP or NADPH. I'll let you enjoy that. In the cyclic electron flow, electrons cycle back from FD to the, I didn't talk about FD. Anyhow, don't worry about FD. To the PS01 reaction center. The cycle electron flows uses only photosystem 1 and produces ATP, but not NADPH. I talked about that also. No oxygen is released, okay, at this stage, at system 1. Okay, system 1, there is no oxygen being released, right? That's another thing. Make sure you know that. Okay. Comparison of chemiosmosis in chloroplast and mitochondria, very important concept. You should know that. During the days that we used to have essay question, I would add that would be a nice essay question. Compare these two, mitochondria and chloroplast, the chemiosmosis. Okay, chloroplast and mitochondria generate ATP by chemiosmosis, but use different source of energy, of course. Okay, mitochondria transfer chemical energy from food to ATP. Chloroplast transfer light into the chemical of the energy to make ATP. Spatial organization of chemiosmosis differ between the chloroplast and mitochondria also shows the similarities. In mitochondria, protons are pumped to the intermediate space of the ATP synthesis, the ATPase, and then synthesis occurs in chloroplast. However, protons are pumped into the thylakoid space and drive ATP synthesis as they diffuse back into the stroma. Here they are showing you the differences between ATP, chloroplast, and mitochondria, how they go ahead through the ATPase. This is the ATPase channel. And then, of course, where the electrons and everything else comes from. ATP and NADPH are produced at a side-facing stroma where the Calvin cycle takes place. That ATP and NADPH is made. Why chloroplast makes them? Okay, they are needed in Calvin cycle. Okay, are you guys ready? Shall we go ahead and talk about that? So ATP increases potential energy of electrons and moving from, okay, right here. He is explaining on this diagram. I should put my, see what he's trying to explain in this diagram. Photosystem 1, I talked about all of this. He's going into more detail of what is going on in here. But we talked about this, that ATPase is the molecule that you get the electrons, the hydrogen atoms from this in order to occur. And the thing is, all of this is good. We already talked about it, but the thing that this diagram is showing you that ATP and NADPH are going into Calvin cycle. They are needed for Calvin cycle. Let's see what happens. Again, details of that diagram. Details, the FD, it's just a carrier of protons to NADP reductase. Okay, I'm not too crazy about it. Your textbook is making some fuss about it. Okay, the Calvin cycle uses the chemical energy of, here we go, NADPH and ATP to reduce carbon dioxide to sugar. So the Calvin cycle, like citric acid cycle, regenerates the starting materials after molecules enter and leave the cycle. The cycle builds sugar from smaller molecules So as I said before, in biology, anytime you want to put molecules together, you need ATP, you need energy. So in this case, already inside of a cell, inside of the chloroplast, there is a 3-carbon molecule exists. That's why the most common type of photosynthesis is called C3, because of these 3 carbon already exist. And then you want to add another carbon to make it C4, right? ATP is needed. You want to make another carbon, ATP is needed, energy needed, I'm going to say. And eventually you have 6 carbon attached to each other. The precursor was 3 carbon, already existed. And then you have 6 carbon. Those 6 carbon need energy because the cell needs to put the molecules together. Okay, so let's see what the diagram. Carbon enters the cycle. Carbon. Where do these? There is a carbon precursors in a chloroplast. This carbon comes from atmosphere, carbon dioxide. Another carbon comes from the atmosphere. Another carbon comes from the atmosphere. That's what he's saying. So carbons enter the cycle as carbon dioxide and leave as sugar molecule named glyceraldehyde 3-phosphate, G3P. For net synthesis of 1 G3P, the cycle must take up 3 times fixing, carbon fixation. I threw that term at you before. So carbon fixation happens at Calvin cycle. 3 molecules of carbon dioxide. I already showed you that with my fingers. The Calvin cycle has 3 phases: carbon fixation, reduction, and regeneration of carbon dioxide, accepted that RUBP. That's what he's talking about, RUBP. That's what the video talked about that as well. Okay, here's the cycle again. They're showing you the cycle. Carbon dioxide is entering the Calvin cycle, we talked about it, and at the end, you already in here, in Calvin cycle, there is a 3-carbon, and he's going to show you the details. There is a 3-carbon precursor, another carbon comes in and becomes 4, another carbon comes in, becomes 5, another carbon comes in, becomes 6, okay? And then that glucose molecule leaves the chloroplast. So sugar production in C4 plants, that's what I talked about, was C3 occurs in 3 steps. The production, again, these details, I don't expect you to know them, PEP, carboxylase, and all of that, C4, but again, as long as you know there are 3 different types of photosynthesis, I talked about 2 of them, C3, C4, and the next one is CAM, acid acid metabolism. Yeah, this is pretty good, showing you C4 plants and what is happening in C4 plants. Since Industrial Revolution in 1900, CO2 level have risen, and increasing the level of carbon dioxide affects C3 and C4 plant differently, perhaps changing the relative abundance of these species. But anyhow, the effects of nowadays, we have fire in California, so that could change it. Okay, the third type of photosynthesis is crassolucent acid metabolism, CAM. Okay, that would be in jade plants, aloe vera, something that you know, pineapple, I think it's a CAM plant. So these are all CAM plants, and they open up their stomata at night, incorporating carbon dioxide into organic molecules, and stomata closes during day, so the CO2 is released and organic acids is being used in the carbon cycle. Yeah. Okay, sugarcane, pineapple, C4, CAM is pineapples, sugarcane is C4, and it's showing you, if you guys need to know the difference between the 2, here they are, you can enjoy it. C3 plants, these are my notes, not from your textbook notes. C3 plants are plants which uses carbon dioxide directly from the air. Soybean, rice, make sure you know these examples. Okay, and one dry day, stomata closes to reduce water loss, and C4 plants, stomata close at most of the time to save water, and the enzymes fix 4C compound instead of 3C compound. Plant the C4 plants. Okay, examples of corn. Corn, sorghum, and sugarcane. These are all examples of C4 plants. CAM plants, stomata, as you already know, admitting at night, the Calvin cycle during day, and CAM stands for crassolucent acid metabolism. That's what CAM stands for. Pineapple, many cacti, and succulent plants like juicy jade plants are already talked about. Importance of photosynthesis. Photosynthesis, as I let you read it, it is important. It stores energy for us on planet Earth, and we use all the way from eating food to making buildings from this process of photosynthesis. Right here, he's talking about it and so on and so forth. In the Calvin cycle and right reaction is comparing them, make sure you know them. The greenhouse effect is the last thing I want to mention in this chapter. I don't want to go it over a lot of minutes. So some gases, that's what we are doing nowadays on planet Earth. We are releasing these gases to the atmosphere, carbon dioxide, by driving cars, nitrous oxide factories, methane factories. And then what happens? The atmosphere is trapping these gases. So these gases cannot get out of the atmosphere. They stay in the atmosphere, okay? And then what happens when the sun sends these light rays, it also has heat rays as well. It's just not the light waves. It is heat coming with those lights. So before the Industrial Revolution, you know, it was the air in the atmosphere. These gases were not here as much, okay? So the heat escaped, went back. Now, these gases are here, does not allow heat to reflect back the earth and get out of the earth. So the heat stays on the planet Earth. I hope I explained it to you guys. So increased use of fossil fuels and gas, photosynthesis counteract the accumulation of course, photosynthesis, the accumulation of the carbon dioxide and all of that in the air, counteract it. But we are cutting forests, we are doing a double whammy. We are putting too much carbon dioxide into the Earth atmosphere. And also we are cutting trees. The trees that they counteract this accumulation of carbon dioxide in the atmosphere. So we are left with a lot of carbon dioxide in the atmosphere, and the heat waves comes from sun, but it cannot go back, escape the earth. I think that is make connections between them. Please study these things in your studies. Make sure, of course, as you know, chloroplast and mitochondria have their own DNA, and this is showing you some of the things that are being made and so on and so forth. Right, I'm gonna go ahead and stop recording. Sorry, I'm getting all sort of messages here. Stop recording.